Hi, I'm Avi Gupta.
And I'm Jai Gupta.
We are currently juniors at Seminole High School in Florida. Today we would like to share with you our research combating antibiotic resistance by using natural compounds as down-regulators of ESKAPE pathogens biofilm formation. According to the scientific journal Nature, antibiotic resistance will take up to nearly 40 million lives by 2050, highlighting the urgent need for enhanced therapeutics to combat it.
Humans have used antibiotics to fight diseases ever since they discovered that the mold on bread can be used to fight their infections 2,000 years ago. Since then, antibiotics have saved millions of lives and have upheld the agriculture and farming industry. However, in recent years, antibiotics are being overused for prophylactic use in hospitals to agriculture. Bacteria possess the intrinsic ability to evolve rapidly because of their high mutation rate. This has led to the emergence of multi-drug resistant superbugs, which most antibiotics currently are not effective against. The current method researchers are using to combat this is to come up with new antibiotics to get ahead of the evolving superbugs. This method, however, can take up to $1.5 billion and takes up to 10 to 12 years to make.
We hypothesized that instead of this traditional arms race between the bacteria and the antibiotics, we introduce a new component that would work together with existing antibiotics to make them effective again. Essentially, rather than starting from scratch and creating a new antibiotic with a new means of inhibition, we take existing antibiotics and combine them with natural compounds, such as those from tree bark or algae. This is one of our molecules we used, apigenin. Antibiotic-resistant bacteria have several methods that they use to circumvent antibiotics. For example, they can use drug efflux, the alteration of identification proteins, and many others. The means that we chose to combat is that of biofilms. Biofilms are groups of sessile bacteria which adhere to abiotic and biotic surfaces with the help of an EPS, or extracellular polymeric substance, as well as a glycocalyx. They essentially pose a barrier preventing the antibiotics from reaching the bacteria in effective concentrations and thus being nullified. This model represents a biofilm covering a bacteria. A biofilm's EPS has several different structures, including polysaccharides, lipids, proteins, eDNA, and more. We are specifically targeting the protein component of the biofilms. This is because they are the easiest to disrupt using shape changes caused by ligands. We targeted ESKAPE pathogens, a group of 6 highly virulent bacteria known for their ability to fend off antibiotic treatments, as well as their tendency to form biofilms. We decided to conduct our study on ligands isolated from natural sources, in hopes of finding if they had any potential in combination therapy, along with antibiotics like erythromycin, destroying the biofilm and allowing the antibiotics to serve their purpose.
Here's how it works. The ESKAPE pathogens are able to protect themselves from antibiotics by using a protective layer called a biofilm. This biofilm stops the antibiotics from reaching the bacteria. Our solution is to use natural compounds to disrupt the biofilm and allow the antibiotics to destroy the bacteria. Our research was split into 2 parts: the in silico research we did and the lab work we used as a proof of concept. Our in silico portion was split into 6 steps to identify effective combinations between ligand molecules and the biofilm proteins. Using the NCBI database, we identified 10 proteins found in the biofilms of the pathogens we targeted, such as AG43 or PA5033. Using the Swiss model and Fire2 software, we modeled these proteins so we could look at the combinations between them and a ligand. Through our online research, we identified 23 potential candidates that can be used to disrupt the biofilm protein, such as bicalin or epicatechin. Using Swiss ADME, we tested the pharmacokinetic properties and drug-like nature of these ligands in order to see how they interact in the body. Using the Swiss doc software, we assessed the effectiveness of ligands on biofilm proteins by testing the binding affinities of many hundreds of combinations. This step took the majority of our time, as we had to manually find which combinations were best based on the amino acids in the protein and the likelihood of it occurring. Lastly, we tested the toxicity of these ligands using ProTox2, which can tell us the potential effects on the body.
For this in vitro portion of our research, we conducted our work in a lab in Kolkata, India. We did this at the University of Engineering and Management, under the supervision of Dr. Dibyajit Larry. We started by spending 5 days culturing the biofilms of Staphylococcus aureus and Pseudomonas aeruginosa, while also testing further in silico research on the specific combination between curcumin, which we would be using as our proof of concept. The next few days were spent conducting minimum inhibitory concentration tests, or MICs, for several trials of each combinations of curcumin, our selected ligand, along with erythromycin, to downregulate the biofilm-forming bacteria which we had cultured. Each day, we would set up one round of MICs, while analyzing the results of the previous day, over and over again. In total, we ran several trials for the concentrations, incrementing by 5s until we reached 30 micrograms per milliliter for each. In afternoons, we would conduct supplementary testing, including analysis of the EPS's composition, as well as examining at a deeper level the interactions of our ligands underneath a scanning electron microscope. This allowed us to visualize the impacts at a molecular scale and for us to literally see the results of our work.
Our in silico work showed conclusive results with some combinations of ligands and proteins having binding energies less than -8. This high binding affinity shows good potential as a therapeutic in the future. Some molecules such as bicalin, hyperforin, and curcumin showed consistent inhibitory results over several proteins. Our wet lab work proved the capabilities of molecules such as curcumin by showing that it can decrease the protein concentration and nucleic acid content in the biofilms. This acts as a proof of concept, showing that our in silico work can accurately predict the effects of ligands on the biofilm. Of course, further testing on each drug with the combination of bacteria and antibiotics must be conducted before bringing any product to the market. However, our in silico and in vitro results showed the potential for at least one combination to act as a life-saving therapeutic. In conclusion, we see these therapeutics having high potential as a combination therapy with other antibiotics as a cheaper and more effective solution against this antibiotic resistance crisis.